Welcome to a Taconic T-Onco webinar entitled Oncology Drug Delivery in Preclinical Models, PK-PD Relationships, given by Dr. Francis Bichot from our French oncology partner company, Oncodesign.
So here is Francis speaking. Thanks to people attending this talk today entitled Oncology Drug Delivery in Preclinical Models, Pharmacokinetic and Pharmacodynamic Relationships. Good morning for US citizen and good afternoon for European one. Next slide, please. So this overview describes briefly the summary of this webinar today. I will start with a short introduction and then a definition of proof of concept study in preclinical oncology, where I will then detail the optimization of tumor drug exposure by different drug delivery systems. Some examples using osmotic pumps delivery will be described in mice. Then long-term drug exposure will be also explained after IV infusion in nude rats. And then I will detail one example of optimization of treatment schedule again in nude rats after IV infusion. A slow release formulation with approved drug will be detailed in the presentation, and a specific tumor targeting using SPECT imaging to finish by a summary of pharmacokinetic pharmacodynamic relationships in one study before conclusions. So first for introduction, why drug exposure is so important in preclinical studies. For sure, the preclinical development of new drugs in oncology needs in vivo proof of concept studies where we have to demonstrate the expected effect of the drug based on its mechanism of action, and if possible to detect and manage the adverse side effects as early as possible in these preclinical steps. The objective is to deliver the right concentration of active drugs at the tumor. So, as a basic representation here, I draw arbitrary concentration of drug after a bolus injection repeated every day, where we observe here. Trouble here. So we observe a very high concentration when the drug is injected after bolus injection, and a very short half-life of the drug inside the body. While when dosed continuously, the concentration of drug remains constant during the duration of the exposure. For sure, from this opposite PK profiles, we have to define probably the best proof of concept study and its main criteria that I will describe in a couple of slides. So, to the right question, we need the right answer. So, does we want to optimize the treatment schedule, either the dose, the route, or the repeated injection or a single injection? Thus, we have to define a therapeutic index to find the lower efficacious dose versus the higher efficacious dose without any toxicity. Thus, we have to establish pharmacokinetic pharmacodynamic relationships when we have to establish relation between the concentration of drug with its effect, whatever the effect. Thus, we have to determine the biological active dose. It's quite now true for targeted therapy. Thus, we have to define a rationale for combinations when we have to perform one treatment compared to another one, how we have to combine the drugs, at which dose we have to combine the drug, and also how we have to reduce the toxic adverse side effect of drugs before regulatory toxicological study. So the global idea is to build a rationale for clinical design. So The main point for the proof of concept study came first from the drug. Here we speak about the new chemical entities. The molecular weight is quite low. It could be peptide as a small agonist, antagonist to receptor, but also biologics as a nucleic acid or antibody. For sure, the design depends also on the mechanism of action of the drug. Is it a targeting agent? Does the mutation need to be present inside the tumor cells? Is it an anti-angiogenic agent which targets stromal cells or endothelial cells? And in that case, we have to consider the murine component and not only the human component inside the tumor. Is it a sensitizing agent which need to be combined? And in that case, we have to consider the sequence or the treatment schedule as very important. And for sure, does the model already express the target? Meaning that we have to know, if possible, if previous in vitro assays. Here is an example of the response of one tumor cell lines to docetaxel, where we have a dose response of the cell lines towards these drugs. And then we have to consider this concentration of drug here, about 1 nanomolar, as very important to reach in vivo to observe the expected effect of the drug. Does the target is still present on the tumor cell? Here is an example of the CXCR4 expression in Ramos cell lines, which helps to choose the cells, or does the target is expressed as shown here by immunohistochemistry for HER2 in tumors from mice. So the physical and chemical properties of the drug are quite also very important. It depends on size, solubility, and it's quite a condition for bioavailability. The drug should be either in a solution for IV injection or suspension for oral route. The solution should be stable during the duration of administration. Any precipitation should be avoided. Any degradation or interaction with plastic should be avoided and known before designing the proof of concept study. For sure, if any information about pharmacokinetic and biodistribution are known, it's quite better to consider oral absorption, to consider metabolism in the different strain of rodent we would like to use. And we have also to select the rodents and the strain, either mouse or rats, depending on the tumor we would like to investigate between the murine or human tumors. And also keeping in mind that the quantity of drug needed for rats is quite about tenfold increase compared to mouse. And for sure, we have to consider, based on the mechanism of action of the immunocompetent or immunodeficient strain of this rodent. For comparison, mouse is quite well known. It's a very small animal, easily available, not expensive, not too much expensive, well documented since most of proof of concept studies in vivo are done in mice. With a good immunotolerance, it's used for compound screening, therapeutic index definition, proof of concept, but we know that mice has a very, very active metabolism, inversely proportional with the size of the animals. In contrast with rats, which is quite robust, he has a longer lifespan, a larger size, and easier for surgery. It's quite possible to choose all kind of administration routes. It's quite possible to do repeating sampling. The same strain will be used in general for regulatory toxicology studies. The dose from our experience is closer to man compared to mouse, and because the ratio between mouse and rat is about 5 to 10. It's quite possible for surgery reasons to have orthotopic models to establish pharmacokinetic pharmacodynamic relationship, to perform imaging, and it's for us the real and well-known pharmacological bridge. So we have to consider as soon as possible the different routes of administration we would like to investigate. So for sure the intravenous, oral, intraperitoneal, intramuscular, intrarectal, intratumoral, or intravesical routes. But as alternative, we also use a drug control release infusion using micropumps, where the drug is still filling the pump and released during a defined time frame, either 7 days to 28 days. But another alternative is also to have a drug control release infusion via the venous system. Here is an example in the picture of mouse already infused by the jugular vein in mouse. So, but the same could be done using the femoral vein in rats to maintain the treatment during very long time. I will describe then this kind of result in few slides. And for the proof of concept study design, we have to consider pharmacodynamic readouts. For sure, the basic ones are body weight, tumor volume, or survival, but also we have to consider proteomic, genomic, or imaging biomarker which could be added and could have value for to design then clinical trials if we can establish this one as early as possible. So let's move forward in the different example. First of one is the use of osmotic pump delivery in mice. The first example was done with ovarian NIH OFCAR3 ascites in mice injected in the peritoneal cavity. The treatment started at D1, and the survival was used as readout. The drug was done by infusion through micropumps during 2 consecutive months, and the pumps were replaced after 1 month. The control was the mice were dosed with Taxol as standard of care, and the Taxol was dosed once a week for 3 consecutive weeks. The arrows are still mentioned here. This figure represents the body weight of animals. And we see a strong increase of the body weight of vehicle and drug-treated animals. The body weight increase is due to the development of the tumor inside the peritoneal cavity, meaning that the mice were sacrificed during this period. In contrast with Taxol and drug plus Taxol, where we did not see any strong body weight gain. The results as survival are still presented here. As described before, the mean survival time of the control was about 40 days, while for Taxol alone, we increased by about 3 times the survival at about 100 days, and we increased again by a factor of 2 the survival of mice treated with drug already done by osmotic pumps and Taxol, meaning that this sensitizing drugs is able to improve the survival inside this model when dosed continuously during 2 months. So here is another example using osmotic pumps where the drug as a peptide was not active when it was dosed by systemic routes. We used one orthotopic U87MG glioma model in mice, and the mice were randomized when the tumor grew inside the brain by using magnetic resonance imaging. The treatment starts based on their tumor volume when the tumor reaches about 1.2 cubic millimeter, and we deliver intratumorally the drug using a brain infusion kit during using a 7-day release apparatus. And temozolomide was put in as a control and positive control groups, and tumor volume was the readout already measured as before using MRI. So here is an example here of mice with a hole inside the tumor where the drug was infused. And here is the complete infusion process inside the brain and inside the tumor of the mice to deliver the drug. First, here is an example of mice from the vehicle groups randomized at T0. So the size, we've seen here the tumor inside the brain, and the tumor grew about 10 days later at this size for this vehicle. For mice infused by the drug through the micropump during 7 days, the tumor remained quite stable during the time course of the treatment, since at the end the size of the tumor here was comparable to the size of the same tumor 10 days before. And as positive control, temozolomide did the same when dosed orally at 10 mg/kg every day during 5 consecutive days. The size of the tumor remained stable during the time course of the study. Here is a full comparison of the tumor volume measured by MRI, where at randomization D17, the size of the tumor were quite comparable, and it increased in the vehicle groups here 10 days later, while they stay quite at the same level for drugs infused directly inside the tumor, but also for temozolomide as positive control. So this data had a great value to establish a rationale for drug positioning in clinic, where the drug was evaluated locally after intratumoral treatment in men. So the next couple of examples is named a long drug exposure. It was done in rats. And I have to thank Dr. Robert Copeland from Epizyme teams, since the data were only present during the last AACR presentation in Washington. And thanks again to Epizyme to be okay to present this data today. So we used during this study the drug named Epizyme 5676. This drug is a small histone methyltransferase inhibitor targeting DOT1L complex, and the study aimed to demonstrate a significant and dose-dependent anti-tumor activity of the drug. For that, we used MV4-11 acute myeloid leukemia subcutaneously grafted in nude rats, and the drug was infused continuously at 35 and 70 mg/kg by day during 21 consecutive days, meaning that the rats bear a catheter inside the femoral vein, and the drug was infused continuously during 21 days. During the infusion, the blood was collected from the rats, and tumor volume was used as readout. So here is the result showing the tumor volume over the time. So the control grew quite fast because we have to perform a sacrifice within about 1 month. And we observed a moderate but significant response of the drug at 35 mg/kg by day, while we observed a complete regression of the tumor over the time when the rats were dosed at 70 mg/kg by day. And to complete the pharmacodynamic data, we collect the rat every week to check the drug concentration in plasma. And here is summarized in this figure, where we observe that the drug concentration over the time was quite stable at both doses of 35 and 70 mg/kg by day. And we established that the plasma drug concentration was about 180 for those of 35 and about 340 for those of 70 mg/kg. For sure, these data were quite very potent and nice to move forward for Epizyme projects. The next example of tumor-bearing rat already infused by drug was also done to optimize the treatment schedule of drug. And here I speak about one drug named 3N2755, and the mechanism of action of the drug is to do alkylating in protein and nucleic acid. And the drug demonstrated previously significant anti-tumor activity in tumor-bearing mice when dosed IP. And the aim of studies here was to demonstrate a significant and dose-dependent anti-tumor activity of the drug after IV infusion dosing. So we used here tumor-bearing rats, and the tumor was MDMB-231 as a human breast carcinoma. The drug was dosed twice a week by IV bolus or 4-hour infusion. So it means that we've put a catheter inside the femoral vein in order to infuse during 4 hours the drug twice a week. And we have tested a range of doses for both of these routes of administration, and tumor volume was used as readouts. So first, by IV bolus twice a week during 4 consecutive weeks, we observed a significant increase of body weight of rats when they were treated with the drug at between 160 to 360. And similarly, after treatment by IV infusion during 4 hours, we observed a significant increase of the body weight of rats when the drug was dosed between 30 to 240 mg/kg. So we observed an acceptable safety profile of the drug on body weight. And by IV bolus, we observed here the tumor volume over the time. In blue, the control grew quite nice during about 1 month, and we observe a complete regression of tumors from all groups treated between 160 to 360. We observed a marked tumor regression for all doses. When dosed by IV infusion, we observed a dose response in this model where the dose of 30 was not efficacious. We observed a moderate activity at 80, while a complete regression of tumor when dosed at 240, like by IV bolus. So we observed this significant dose-dependent anti-tumor activity after infusion, which was not shown before. And at the end of the study, we did the collection of tumor, harvest the tumor, proceed to mechanically and enzymically description of the tumors to perform ex vivo colony assays, and so to check the viability and the potency of the remaining cells to form colony. And we observe for all groups that the tumor cells have lost a part of their potency to form colonies. It was very strong for IV bolus at 360, but also strong for the other doses and routes of administration. So the in vivo treatment leads to reduce the potency of residual tumor cells using this assay. In parallel, we also collect healthy tissues like liver, kidneys, and bone marrow to proceed to histology analysis and compare to control the drug at 240 mg/kg done by IV infusion did not change significantly the histology of these healthy tissues since they remain normal. And we also checked by blood numeration the formulation, where we studied the white blood cells, red blood cells, and platelets, and we did not see any strong difference of these blood cells compared to control, except platelets for one group here. So the blood numeration remained normal, and based on this data, it was decided to combine the drug with CPT11 as topoisomerase 1 inhibitor. So here is in blue the curve of the tumor volume over the time of the control. CPT11 alone was not too potent in the model. The drug by itself alone was very potent, as shown before, at 240 already IV infused during 4 hours. And we observe a synergy when combined with CPT11 here, since the T/C value was about 9% compared to 24% of the drug alone. So again, to conclude, no toxic adverse side effect of the drug was observed at the biological active dose. A dose-dependent anti-tumor activity of the drug was observed after 4-hour infusion in tumor-bearing nude rats. The drug has entered in phase 1 clinical trials, and it was dosed during as a slow IV infusion during 4 hours. And to complete the story, DNA adducts in lymphocytes were measured as a PD endpoints after treatment in the clinical trials. So next step, move forward to the slow release formulation. Very good one. So here I will speak about Degarelix. Degarelix is now an approved drug since it's a gonadotropin release hormone named GnRH antagonist. This drug is a peptide which forms a spontaneous gel when injected subcutaneously and leads to sustained release of drug in the bloodstream. And the study aim was to compare Degarelix with Detripsis LHLH super agonist, since this drug was approved previously on the market. And as a PD endpoint, we studied the plasma testosterone suppression, the PD profile on organs, and for sure the anti-tumor activity. We use for this drug a very nice model named R3327H rat androgen-dependent prostate model in Copenhagen rats, and the model is also named the Dunning model. So we performed a short-term experiment during about 1 month, where the drug was dosed subcutaneously at 1 mg/kg once a month, and we use, for comparison purposes, Detripsis dosed subcutaneously at 0.5 and 1 mg/kg once a day. And we also performed a long-term experiment during about 9 to 10 months, where the drug was dosed also at 1 mg/kg once a month, and we used leprolide depot as the clinical form of the Detripsis dosed subcutaneously at 1.5 mg/kg and dosed every 3 weeks. During this study, we collect tumors, tissues, and blood, and body weight, tumor volume, GnRH expression, and plasma testosterone were used as PD readouts. So first, about the PK. Degarelix PK parameters confirm the sustained release of the formulation. It means that the Cmax were quite low, but it depends on the dose already administered between 0.05 to 2 milligram by kilogram. The Tmax also was quite short, between 1 to 24 hours after the single injection. But as expected, the half-life of the drug reached about 8 days here for the dose of 0.2 to 40 days when the drug was dosed at 2 days. So after this confirmation, we checked the effect of Degarelix on plasma testosterone. So first, this figure represents the testosterone level in rats over the time. So in black, we have the control here, where the mean value is about 1,000 picogram milliliter. For sure, in castrated rats, there is no remaining testosterone. And for the detripsis, we observe first a strong, we observed a flare-up period. This flare-up period corresponds to a burst of testosterone after injection, which also occurred in human, and it increased a lot at 20,000 picogram milliliter. For degarelix, the story is different, since the testosterone level in rats was decreased very, very quickly and remained at zero during the whole course of the study, meaning that degarelix suppressed completely the plasma testosterone level just after injection, in contrast with detripsis. During the same time, we also measured the tumors. So in black, the controls, the normal growth of these slow-growing prostate tumors, and in blue, the degarelix curve, where which show that the tumors did not grow after treatment, and the curves look like those obtained with castration. Interestingly, we observed during the first time a similar growth of the detripsis-treated groups compared to controls, probably due to the flare-up period and the burst of the testosterone level in those rats compared to the others. And then in a second phase, we observe a response here of the tumors to detripsis. So in a long-term experiment, again, degarelix suppressed completely the plasma testosterone level. Here, the results of testosterone are expressed in nanogram milliliter, and so we observe that, and the time points were not the same. The rats were only collected every 2 weeks. And we've seen after degarelix that the concentration of testosterone was decreased at zero, like castration, and it was slightly delayed for leporide depot. And in parallel, we observed a strong anti-tumor activity of degarelix at 1 kilo once a month, and the growth curve looks like those obtained with castration, in contrast with leporide depot, since the growth of the tumor restarts again about 150 days after the treatment starts. So here we've demonstrated that degarelix has the same effect compared to castration, and better than leporide. At the end of the short-term experiment, we removed the tumor and testis to weigh this organ, and we've shown that for both tumors and testis, the degarelix decreased their weights at the end of the study, and it was also the same for detripsis at a lesser extent. For sure, the tumors size were quite comparable between castrated and degarelix-treated rats. Similarly, we have collected prostate and seminal vesicles at the end of this short-term experiment, and in both cases we observe a strong decrease of these LC tissues in both detripsis and degarelix-treated groups. So also at the end, we perform the collection of pituitary glands from the rats in order to analyze the number of mRNA from GnRH receptor. And we've shown here that degarelix suppress strongly the level of GnRH mRNA. It was also at a lesser extent the case for detripsis. And the drug by itself probably induced a profound reduction of luteinizing hormones, LH, and follicular stimulating hormones, FSH, which turns to a strong plasma testosterone level reduction. So the Biologics demonstrate the sustained inhibition of the tumor growth, at least comparable to surgical castration. It suppresses the tumor growth, plasma testosterone, and LHRH in tumor-bearing rats with a faster onset of action than leprolide. And as mentioned before, the Biologics slow-release formulation was approved for advanced hormone-dependent prostate cancer about 5 years ago in both US and Europe. So I cannot finish without any example of a specific tumor targeting and without any imaging data. So here is an example here where we studied trastuzumab as antibody, as a HER2 targeting antibody, and but this kind of application was also done with peptide and others. So the idea is to label the drug. Here this antibody is with radionuclide indium-111 to do a biological validation of this new labeled chemical entity. And the study aim was to correlate the biodistribution of this antibody with its anti-tumor activity. So for sure, first we have to consider multi-step synthesis and radiochemistry of the drug. Here is the antibody already coupled with a chelatant named DOTA in order to reach here antibody coupled with DOTA. And in a second step, indium-111 was added in the chelatant to have the full radiolabeled antibody to be used then for imaging and biodistribution purposes. So first, we checked by FACS analysis and saturation binding experiments the potency of this new radiolabeled entity to bind HER2 expressing cells. By FACS first, we checked that the labeled antibody named MM343 binds this HER2 positive cells similarly to the original trastuzumab. And is in saturation binding experiment, we also checked a specific binding here of this new labeled entity in HER2 expressing cells. We know before that the trastuzumab displayed a significant anti-tumor activity in one HER2 expressing tumor model in mice, and we have chosen a non-saturating dose of the radiolabeled antibody at 1 mg/kg to do the imaging using indium-111 trastuzumab antibody. So. And then we check the specific tumor targeting using this radiolabeled antibody in this model, where using a SPECT CT machine, so 24-hour post-injection. Here is the picture of the mouse where we have the CT scan here with the bone, with the spinal cord here, compute with the images from the SPECT, which shows the body of the mice and the tumor, which stain the indium-111 corresponding to a specific tumor targeting. And over the time, for same animals, we've maintained this strong staining of the tumor by the indium-111 antibody. Similarly, at the end of the study, we sacrifice the animals to check the specific tumor targeting, and so we've shown that the tumor contained a high percentage of the injected dose here, about 60%, while it was quite low in blood, liver, and kidneys, and shown before using SPECT imaging in mice. And in parallel, we used this LC tissue to check the binding of the indium-111 antibody, again tumors, but also different tissues like kidneys, lungs, and liver by autoradiography. So this technique is quite very useful for antibody-drug conjugate optimization and selection of the right vector. It's quite possible to perform this kind of specific tumor targeting by SPECT, but also by PET imaging, as we did before. So to conclude and to have a large idea of PK-PD relationships, here is the last example I found from our work here in a very, very big study performed a few years ago. In this study, we use diflumotecan, named also BN80915. This drug is a topo-1 inhibitor. It was dosed by a 10-minute IV infusion by the tail vein to tumor-bearing rats at 1 milligram kilo. We consider for this study about 200 rats. It was a 2XXL size to perform PK, biodistribution, PD. During the course of the study, the treatment were repeated every week, and we've monitored body weight, tumor size, we've collect blood and tumors to establish PK biodistribution. So first, we observed that the PK profile of the drug was quite similar after infusions every week, since the PK profile here are similar. Then we also consider the different PK and biodistribution of the drug inside the body of the rats, where we've shown here that the drug is quite eliminated quite rapidly from the blood and maintained about 2 times better in the tumors. And we've seen also here that the main excreting organs were both kidneys and liver. We also performed we also performed hematological follow-up on the same rats, where we observe a strong decrease of the white blood cells and a strong recovery of these white blood cells after the end of treatment. In parallel, we observed a significant anti-tumor activity of diflumotecan, since the growth of the tumor here was lower than the growth of the control. We studied in subcut tumor the mechanism of action of the drug by detecting the DNA topoisomerase 1 cleavable complex here in tumor treated by diflumotecan, and this DTCC complex appeared within 1 hour after treatment and disappeared slowly after treatment within the 2 days. So taken all together, this data was very useful to establish PK-PD relationships in preclinical, and this treatment schedule was then used in clinic since the drug was dosed during a 20-minute IV infusion in patients, and the DTCC PD readout was also applied in clinic since it was developed for circulating lymphocytes. So as a conclusion, for sure the drug delivery remains a critical step in preclinical development, since it impacts on pharmacokinetic, metabolism, efficacy, drug positioning, and many parameters and alternatives should be considered to improve drug efficacy in cancer. For sure, Oncodesign developed expertise in optimizing the pharmacological parameters of the drug, including new strategies with studies on antibodies, nanoparticles, topical application as a wax, polymers, and so on. So thank you for your attention. For sure, if you have any question, you have my email address, and you have few times to have an online question during the webinar. And I have to mention also that both Taconic and Oncodesign will have a booth during the next World Pharma Congress planned early June in Philadelphia, and one of my colleagues, Jean-François, will talk during this meeting about in vivo complex chimeric NOG mice model development and use of preclinical evaluation of drugs. Thank you very much for your attention.
Thank you for the great talk. We do have a couple of audience questions. Let's see, our first one is, what other PD markers could be interesting to monitor in preclinical experiments before entering the clinic?
Sorry, would you like to repeat, please, Megan?
Sure. Which other PD or which other pharmacodynamic markers could be interesting to monitor in preclinical experiments before entering the clinic?
So, as I mentioned in my talk, all of body weight, tumor volumes, DTCC, PD markers could be used. For sure, in preclinical, some other could be added, like progenitor endothelial cell, circulating endothelial cells, platinum adduct or other carcinogen adducts. We also checked for some of drugs the status of the vasculature of the tumor using different markers like CD31, smooth actins, but also by using fluorescent dye like EUX to have an idea about the functionality of the vessels inside the mice or rats. We checked different parameters for hypoxia like carbonic anhydrase 9. We did some collection of skin, notably for EGFR inhibitors, and for sure a panel of imaging like K-trans parameter by DC, MRI, spectroscopy, also by for MRI with different metabolite content inside the tumor, which could change over the time of treatment.
Okay. Our next question is there some interest to use orthotopic models in mice and rats to optimize drug delivery?
Yeah, I only mentioned here in my talk orthotopic model when the glioma was put in the brain. For sure, there is a real interest in drug discovery. Why? Because drugs are still dosed by IV, IP, and so on, and we know that the vasculature and so the delivery of drug to the tumor could be different from one part in the animals compared to another one. And the orthotopic localization of the tumor probably reflects more the clinical situation, and probably that the drug delivery could be different and more appropriate compared to a standard subcutaneous tumors, where we know that the vasculature is different. Again, we are still speaking here about preclinical models, and we have a different answer from one model to another one, but definitively I think that the orthotopic model have interest towards the drug delivery.
Okay. Our next question is about peptides. Can infusions be done in both mice and rats with peptides?
So we did peptide infusion before in both mice and rats. But I have to say that we had sometimes bad experiences, since the peptides could be charged as a cation or anion, and it also could lead to strong adverse side effects when injected IV or as a bolus. And so the peptide has also sometimes some trouble and some stability issues, precipitation issues, or binding issues in preclinical model, which should be considered when you have to design a proof of concept study.
Okay. Let's see. Are there some alternatives to micropumps that exist for infusions in mice?
Yeah. So you speak about micropump in mice?
Yes, so the question is alternative to that could be used in mice.
I think again at Oncodesign we used a micropump in mice, but it should be done as subcut or IP in order to grab the pump in the mice. We did some experiments and successfully experiments where we've put a catheter in mice in the jugular vein in order to continuously infuse the drug in mice, and then it's quite easier in that case to follow the drug concentration over the time, to check that everything is okay, mouse to mouse over the time.
Okay, the next question is about the transition from preclinical to clinical. So how do you translate the schedules from preclinical studies to clinical trial schedules, particularly when infusions are used in the preclinical studies?
So, as mentioned in the talk, both diflomotecan and 2755 drugs were applied in early phase 1 clinical trial in the same treatment schedule defined in tumor-bearing nude rats. It was 4 hours once every 3 weeks for 3N2755, and it was a 20-minute infusion for diflomotecan compared to 10 minutes we did in nude rats. So my feeling is that we have to consider this preclinical experiment as a strong rationale for the clinical trials.
Okay. And this will be our last question.
Yep.
About rat versus mouse tumor models. Which one should a researcher choose? Do you have any general considerations that you can speak to?
Very strong, very difficult question. I'm quite not sure about the question. Does we speak here about the difference between rat and mouse, or does we speak here about the tumor by itself coming from rat or mouse? I will try to answer both. For sure. The choice of the strain to perform preclinical studies clearly depend on the drug, availability of drug, and depend on at which step we have to consider this preclinical study. In very early stage, I would suggest to first use the mouse in order to demonstrate as early as possible with low quantity of drug the potency of the drug. When we have some evidence in mice of the potency of the drug, I think that the treatment schedule, the doses, the route of drug administration should be considered in rats, because we have access with tumor-bearing rats at many events coming from blood collection, coming from imaging results, and so on. So this is my consideration based on our experience. And for sure, as I mentioned during the talk, in oncology, in general, we reduce the dose in rats by 5 to 10 compared to mouse, meaning that, and to have the same anti-tumor effect. So, and this dose is probably more close than from the dose which will be used in human. I think that the question was not to choose the tumor between the rat and mouse, but to be honest, we have a panel of both rat tumors and mouse tumors coming from chemo-induced models or coming from very strong and very well-known models, very old models. Again, depending on the mechanism of action of the drug, we have to consider the stroma from each host before designing the study design and the proof of concept study.
Okay. Thank you very much for answering those questions, and thank you for a great webinar. Unfortunately, we are out of time. If There are some questions that we did not get to, and we will follow up with those by email. And you will also receive an email with a link to the presentation. Thank you again for attending.